Hi everyone, this is Mr. Hall and this is a quick video about rate of digestion calculations. This is something that kind of creeps in on many sort of schemes of work for biology, and it's in the AQA scheme for those of you that are studying it. And it's not too difficult, but it's something that you do have to know. The good thing is that it's quite similar to the stuff that you need to know for chemistry with rate of reaction, so a lot of the skills you have to know are really similar for the two. So today what I'll go through in order of rate of digestion calculations is how to calculate rate of digestion from data, how to calculate rate of digestion from graphs, and then to describe how the graph for the rate of digestion would change if a different pH or temperature was in place. Now, the thing I won't talk about today is calorimetry, because calorimetry is another technique of working out the rate of digestion, but it's just a lot harder than the standard calculation, so that might need its own video, which I might do at some point. But I'll just focus on the basics for today. So in terms of an equation for rate of digestion, it's basically the amount of nutrient divided by time. Okay, so the nutrient, it can be what's made or what is broken down. Now, most commonly, it will be for what is made. If it's what is broken down, it gets a bit more complicated and you have to do a few different bits and bobs, which I'll go through a little bit later on. But mostly it's what's made. So, for example, if you have a question of like 9.5 grams of protein was digested in 90 seconds, your rate would be 4.5. And then you would divide that by 90. And that would give you, if I get my calculator out, so 4.5 over 90 would be 0.05. So that would be 0.05 grams per second, okay, which is like an astonishing rate of digesting protein. And then another problem would be, let's say you've got 12.5 grams of glucose produced in 20 seconds. That would be 12.5 divided by 20, which would give you 0.625. Again, it's grams per second. A couple of things about these calculations. Number one, always show your working, as you can see that I've done here. I show out in full just in case I make a mistake in the calculation part, and then always show your units. Okay, so as long as you've got those two bits, you're good to go. Okay, so moving on. The other thing that you might have to do is analyze from graphs. Now, graphs is pretty much the same skill, except you have to be able to read data from graphs and then come to a conclusion about what the rate is. Now, this is all about the rate of amino acid production. So we're assuming that protein's been broken down in the body over the course of 18 minutes. I think the 18 is hidden from view because of the image. And what we're going to work out is like what the rate is at different points. So let's say I'm working out the rate at 3 minutes. Okay. Now, to work out the rate at 3 minutes, I have to go all the way up to where the 3 minutes is on the curve, and 3 minutes corresponds to 25 grams. So the rate here would be 25 divided by 3, and that would give me 8.33 grams per second. And then if I did it at 6 minutes, grams per minute, I should say. And if I did it at 6 minutes, I'd go up to the 6 minute point, which is here. And that corresponds to 35. So that's 35 over 6, which would give me 5.83 grams per minute. And let's do it at 12 minutes just for the crack. So at 12 minutes, that's around maybe 41, 42, let's say 41. So it's 41 divided by 12, and that gives me 3.42 grams per minute. Now, the thing you might notice is as the reaction or as the digestion proceeds, the rate goes down. So whenever this kind of experiment is done in a lab in a test tube, the rate of digestion will always decrease. Now, you'll rarely be asked why that's the case, because they're not asking you to know much about the sort of rate of reaction stuff from chemistry, but just be aware that the rate of digestion will decrease over time until it eventually stops. And at this point, you will assume that all of the protein has been digested and you've made the maximum amount of amino acids. And the time at which the digestion finishes, it was when it starts going horizontal, because you're not making any more amino acids. So it's at that point. So the reaction stops at 15 minutes. Now, one thing you might be asked, and this is particularly important when you're talking about the rate of decrease of a nutrient, is what the rate is between two different points. So let's say we're going to work out the rate between 3 to 6 minutes. Now, if we're doing that, we have to find out what the difference in mass is between those 2 points. So, for example, between 3 and 6, 3 is here, whereas 25, and 6 minutes is up here, which is at 35. So the difference in mass between those 2 points is 35 take away 25, and that equals 10. And then the difference in time between these 2 points is 3 and 6. So the difference is 6 take away 3, and that gives me 3. So your calculation actually is 10 divided by 3, which is 3.33 grams per minute. So there's a difference between working out the rate between two different points, which is this type of problem, and the rate at a particular point, which are the problems which I tackled earlier. All right. Now that's a kind of graph that you might get a lot of, the production of something. Probably more commonly is you'll get a graph about the rate of a nutrient being digested. So the mass is going to go down. So we know, for example, from my earlier example that I showed you, that was amino acids being made, and amino acids are produced from proteins, right? That means proteins are being broken down. So this graph is showing me the rate of breakdown of proteins. And if you look at it quite carefully, I'm just going to draw the line of best fit in for you, but it basically looks a little bit like this. Try and do it as straight as I can, and eventually it will level off there. It's not very straight, but that's roughly what you got. Now, the important thing here is I can't really do the same calculations as before, the rate at a particular point. Because, for example, if I'm doing the rate, let's say here, at 15 minutes. Now, the rate at 15 minutes is going to be, well, the mass of protein, which you've got left over, is 2.2. But if I do 2.2 divided by 15, that will give me, I'll just put it in my calculator, a number of 0.147 grams per minute. Now that doesn't actually make as much sense as something that you've produced, so you're very, very unlikely to get problems like that. But what you might be asked to do is work out the rate of digestion between 2 points for a graph like this. So for example, let's say that we're asked to work out the rate between 15 minutes and 25 minutes. Now between these two different points, we have to do the same thing as before. We have to work out the difference in mass, and then we have to work out the difference in time. So at 15 minutes, the mass is 2.2. And at 25 minutes, the mass is here, which is about 0.6. So 2.2 take away 0.6 gives me 1.6. Now the difference in time is between 25 and 15 minutes, which is 10. So my calculation is going to be 1.6 grams divided by 10 minutes, okay? And that will be 0. I think I'm right saying this, just make sure I don't make any clangers. It's 0.16 grams per minute, okay? And that means that in that 10-minute period, you're breaking down protein at a rate of 0.16 grams every minute. And obviously that rate is kind of an average over the 10 minutes. That will change as you change the points at which you're measuring. That sometimes comes up, so just be aware of that. Now, the only other thing I want to mention before I close this video is what happens if you've got different temperatures and pH levels. Now, if you've got certain conditions for digestion, if you change the pH and the temperature, it will change the activity of your enzymes, which are breaking down food, okay? Now, if you have conditions that are less favorable for digestion, the rate would be slower. So, for example, let's say that these reactions are happening at 40 degrees, okay? Which is reasonably good. Now, if I make it less favorable, let's say 60 degrees, where, you know, the enzymes start to denature, or let's say I put it at 20 degrees, where it's going to be a lot slower, what you'll have is just a much, much slower rate of digestion. Now, it might eventually digest all of it, but it's going to be a lot slower, and it will look a little bit like this, and it will take a lot longer to get to the required amount. And then on this graph, it's going to be a lot slower again. So what you're going to have is, let's say it starts at this point, it will start, instead of having, so this is your initial curve, which looked like this, but then on a much slower digestive sort of tract, it will look a bit like this. So it'll take a lot longer to get to 0. Now, let's say you had more favorable conditions. Now, if it's more favorable, that means digestion is going to be quicker. So let's say instead of 40 degrees, let's say the optimum temperature for this enzyme is 37 degrees. Okay, that's a good example. Or if we're talking about pH, so we're talking about amino acids, let's say it was a pH, the most favorable one is like sort of low 1 or 2. So let's say we did it initially at pH of 5. Now, if I changed it to a pH of 1 or 2, which is more favorable for protease to break down amino acids, you can have a much sharper rate. It's going to break it down a lot quicker. So it might be something a bit more like this, where it's a lot sharper and it gets to that point a lot quicker. And then for this graph, similar thing. Okay, if it's a lot quicker, it's going to reach 0 a lot faster. So it might look a bit like that. Okay, you might get that. Comes up sometimes. Just be aware that that's what it looks like. Okay, and that's it. So I hope this video has been useful, and best of luck with your rate of digestion calculations.